Hello everyone, today we will know about the chromosome. So in this video we will know about what are the different types of the chromosome that are present and what is the role of the chromosome in the organisms. So before going to the chromosome we should know something about the chromatin. So you have already heard the term chromosome. You have already heard the term chromatin and the DNA. So what are the difference between this chromosome, chromatin in the DNA? So now get into this. So in the cell cycle, let's suppose this is the cell cycle. So in the cell cycle there are the different types of the phases are present. So the first stage is called as the gap 1 phase, or it is also called as the G1 phase. So that means, and before that another stage is called as the G0 or the quiescent stage. So that means if any cell, if it wants to go for the division, then it transfers from the G0 to the G1. If it doesn't want to divide, then it remains in the G0. So that's why the G0 is called as the quiescent stage. So now if the cell wants to divide, then it left the G0, then it transfers to the G1 and the gap 1. Then after the gap 1 there is a phase is called as the S phase. S for synthesis. So what is the synthesis, or why the name is called as the S phase or the synthesis phase? Because here the what does the it synthesize? It synthesizes the DNA. So that's why it is called as the S phase. So after the S phase, another phase is called as the gap 2 or G2 phase, and after that, that is the main phase, that is called as the main phase or the M phase, or it is called as the mitosis or the meiosis phase, because in this phase actually the cell division occurs. So the gap 1 S phase, gap 2 is called as the interphase, and this is called as the M phase is called as the division phase. So what is the difference between the interphase and the division phase? Because in the interphase or during the interphase, the cell already prepares for the division, but in the interphase there is no division in the cell. The cell divides only in this M phase, but in the interphase it prepares for the division. So that's why it is called the interphase. Okay, so now come to the term the chromosome, chromatin and the DNA. So what is the DNA? So DNA, simply imagine a thread of simply you can imagine like the thread is like this. So if this thread it coiled together one and more, and now it forms the structure like this, and this is called as the chromatin. Okay, so in the chromatin at the middle it is called as the centromere. So that means our centromere, it is also called as the primary constriction that divides these two arms. This is the one arm, this is the another arm, and this is called as the chromatin. So that means in the gap 1 the structure of chromosome, it looks like this, and this is called as the chromatin. Okay, so that means what is the difference between the DNA and the chromatin? That means the single DNA or the one DNA coiled together each other, and it forms the chromatin like structure and looks like this. That means one chromatin, you can say that one chromatin contains one DNA. So that means at this phase, that is, there are 23 DNA are present. So that means the 23 chromatins are present. So in our cell, for example, in the animal cell, the 23 DNA are present, and this is called as the chromatin in the gap 1 phase. Okay. So now in the S phase, the DNA is synthesized. So that means another DNA, that means in that chromosome one DNA is present, and it called together to form this like structure that is called as the chromatin. So in the S phase, one chromosome or the one DNA replicates, and now two DNA are formed. So that means one chromosome again called together to form the chromatin. So now this DNA also called together, and now it forms like this. That means the two chromatins here, two chromatins are formed here. That means in the S phase. So that means in the S phase, two chromatins are present, and now these two chromatins are joined, and it looks like this. So that means this is the first one, this is the second chromatin, they are joined together to form in the S phase. So now this one is called as the sister chromatid to each other. That means this one is called as the chromatid, this one is also called as the chromatid, and these two chromatids are also called as the sister chromatid to each other. So that means during the S phase, the chromatin looks like this. That means in the G1 phase, the chromatin looks like this. That means in that one DNA is present, and from the one DNA it forms one chromatin, and at the middle it is called the centromere. In the S phase, because the DNA is synthesized, that means the DNA replication occurs. That means from one DNA, two DNA are formed, and from the two DNA, obviously two chromatins are formed, and two chromatins are again joined together. So now each one is called as the chromatids. This is the one chromatid, this is the another chromatid, and they are called as the sister chromatids. So that means the chromatin in the S phase, it looks like this, but the chromatin look in the G1 looks like this. And what is the difference between the chromatin present in the gap 1 and the S phase? In the gap 1, the chromatin contains one DNA, but in the S phase, the chromatin contains two DNA. So that means in the cell, for example, in the human cell, at the gap 1 phase, there is the 23 DNA are present, but in the S phase, 23 chromatins are present. 23 chromatins are present, but the DNA are now the 46 DNA. But here the 23 chromatins are present, and the 23 DNA are present, and the structure looks like this. But in the S phase, the chromatin, same number of the chromatin, that means the 23 chromatins, but the 46 DNA, that means the DNA number doubles. So two chromatids are formed, and each are called as the chromatids or the sister chromatids. So in the gap 2 phase, again the same like structure as present here, but in the division phase, the same like structure are present. Look at this structure, this one, and this one. What is the difference between this structure and this structure and this structure? So during the interphase, that means in the S phase and the gap 2 phase, same chromatin structure are present, but this chromatin is present in the thin structure or decondensed structure. But in the M phase or during the division phase, now it is the very thick. So it is also called as the thick or condensed. So this is the difference between the chromatin and the chromosome, and because this thick or the condensed structure of the chromosomes are present, so condensed chromatin are present, now you can say that this is the chromosome. So that means the chromosomes are actually the chromatins, but they are present in the thick or the condensed structure, and now it is called as the chromosome. So that means this is the chromatin. That means the one chromatin contains one DNA. At the S phase and the gap 2 phase, this is also called as the chromatin, but it contains 2 DNA, and these chromatins are present in the very thin, or it is called as the decondensed manner. But after going to the M phase, it is very thick, and it becomes very thick or the condensed structure, and now this is called as the chromosome. So that means what is the difference between the chromosome and the chromatin? The chromosome is the thick and the condensed structure that is present during the M phase. But the chromatin is the structure that is the thin and the decondensed structure that is present in the interphase. And what is the DNA? DNA forms the chromosome. One chromatin, the one DNA forms one chromatin in the gap 1 phase, and in the S phase, because 2 DNA are formed, so one same chromatin, it contains 2 DNA, one contains, or it is called as the chromatids or sister chromatids. So the 2 sister chromatids are formed in the one chromatin, and here also same structure. That means here also one, you can say that now it is the one chromosome, but it contains 2 DNA. This is this one, and this is the another one. This is the one chromatid, this is the another chromatid. These 2 chromatids are called as the sister chromatid. So this is the difference between the chromosome and the chromatin and the DNA. So this is the all about the difference between the chromosome, chromatin and the DNA. So now come to what are the different types of the chromosomes. So whenever I say the chromosome, so you can imagine that that is the chromatin that is present during the M phase or the division phase. So now come to the types of the chromosome. Types of chromosome. So there are the 4 types of the chromosomes are present. One is called as the metaphase. Second one is called as the submetaphase. Third one is called as the acrocentric, or it is also called as the metacentric. You can say that simply it is called as the different types of chromosome. So first one is called as the metacentric. The second one is called as the submetacentric. Third one is called as the acrocentric, and the fourth one is called as the telocentric. So these 4 types of the chromosomes are present on the basis of the presence of the centromere. So look at this structure. They are the different types of the chromosomes. Look at this metacentric chromosomes. So in the metacentric chromosome, this is the centromere, or the centromere is also called as the primary constriction. Primary constriction means here it forms a gap, or it divides these 2 arms of the chromosome. So this is the centromere, and this metaphase or the metacentric chromosome, the centromere is present at the middle. So that means at the metacentric, the centromere is present at the middle of the chromosomes. So that means this is the one arm, this is the another arm of the chromosome. So now because the centromere is present at the middle of the chromosome, these two arms of present, they are of the equal size. Okay, so this is the metacentric chromosome. So next come to the submetacentric chromosome. This is the submetacentric chromosome. So here the centromere displays of the away just after the middle. That means actually at the middle part is the here, but the centromere is present away, some part away of the middle part. So it is the away from middle. So because the centromere is present away from the middle, so now these also two arms are formed, but these two arms are unequal size. Unequal size. So that means one arm is the shorter. So here this one is short and this arm is a very long arm is formed. So always this short arm is called as the P arm and the long arm is called as the Q arm. Okay, so because now the centromere is present away from the middle, two types of the arms are present. One is called as the short arm, another is called as the long arm. The short arm is called as the P arm and the long arm is called as the Q arm. So this is the submetacentric chromosomes. So again another is the acrocentric. This is the acrocentric. So acrocentric means now the centromere displaces near the terminal part. This is the terminal part. So that means a very large arm or the long arm or a Q arm is formed and a very short arm or the P arm is formed due to the acrocentric chromosomes. So another is the telocentric. The telocentric means the centromere transfers exactly to the terminal part. So terminal part. So that means after the transferring the centromere to the terminal part, so now only one arm can be formed and this arm is called as the Q arm. Why? Because already it is very long. So this is the different types of the chromosome on the basis of the presence of the centromere. So that means in the metacentric, the centromere is present at the middle. So after that or because of that, the equal size of the two arms are formed. But in the submetacentric, acrocentric and the telocentric, unequal size of the arm is present. The short arm is called as the P arm, the long arm is called as the Q arm. So in the submetacentric, the centromere is present just away from the middle. So because a P arm and the Q arm is formed, the acrocentric, the centromere is near the terminal, not exactly to the terminal, near the terminal. That means a very long arm is formed and a very short arm is formed. So at the telocentric, the centromere is present at the terminal. That means one arm is formed and that arm is the long arm, or you can say that it is the Q arm is formed. So these are the different types of the chromosome on the basis of the presence of the centromere in the chromosome. So another chromosome is present, that is called as the satellite chromosome or the satellite chromosome, or you can say that SAT chromosome, which is the same, that is the satellite chromosome. So what is the satellite chromosome? One is the chromosome, one is the satellite chromosome. So what is the difference between that? Look at the structure of the chromosome. This is the chromosome, simply chromosome, and look at this structure. And this structure is called as the satellite or the SAT chromosome. So what is the difference between the structure of this chromosome? So in the satellite chromosome, one centromere is present or the primary constriction, which is present also in the chromosome. So the main difference is this structure. So that means this is the one constriction is present in the obviously in the chromosome here also, and that is called the primary constriction. But some chromosome contains another constriction here. So because of that, of the presence of this constriction, one this like structure is present, and this structure is called as the satellite. So in the satellite chromosome, a secondary constriction is present, and this because of this secondary constriction, it forms a satellite, or you can say that a small fragment are formed. Here also some again another fragment is formed, which is looks like the satellite. So that's why in the satellite chromosomes, because of the presence of the secondary constriction, the satellite of the small fragment is formed. So it contains secondary, or the two constrictions are present in the satellite chromosomes. Not all the chromosomes are the satellite chromosomes. Some chromosomes contain satellite. Which chromosomes? So in human being, it is the 13, 14, 15, 21 and the 22 chromosome. So these 5 chromosome forms the satellite or the secondary constriction. So that means these exact 5 chromosomes are called as the SAT chromosomes or the satellite chromosomes. Now come to karyotype. So karyotype. So what is the karyotype? The karyotype is simply the complete set of the chromosome in an organism, which is called as the karyotype. For example, in the human being, 46 chromosomes are present. So these are the 46 chromosomes are present. So 1, 2, 3, 4, 5, 6. So it is the 46 chromosomes. That means the 23 pairs chromosomes are present. So this is the 1, 2, 3 like that. That is the 23 chromosomes are here present. And now because the 23 pairs are present, so like that again another pair of the 23 chromosomes are present, and this is called as the karyotype. That means the complete set of the chromosome of any organism is called as the karyotype. And this is, you can say that this is the karyotype of the human being, because in the human being the 23 pairs of the chromosomes are present. So how the karyotype is formed? So first, in the cell, in the metaphase chromosome. What is the metaphase chromosome? One is called as the cell division, or the during the cell division. One is called as the prophase, another is called as the metaphase, then anaphase, then the telophase. So during the metaphase, the karyotype is formed. The karyotype we can form the karyotype, or the complete set of the chromosome can be photographed. Why it is taken, or why the photograph of the metaphase is formed or the metaphase is taken? Because in the metaphase actually all the chromosomes are present at the middle of the cell, and after that the spindle fibers are joined together. So this is the metaphase plate or the metaphase stage, and during the metaphase we stain the chromosomes, the chromosome, and the specific stain is used, and that is called as the GIMSA, GIEMSA. A GIMSA stain is used to stain or the dye the chromosomes during the metaphase, which is called as the GIMSA. So after the staining by the GIMSA, we can take the photo. We can take the photo, and the photo or this procedure of the taking the photo during the metaphase by the GIMSA stain, and we can say that it is called as the pictomicrograph. Pictomicrograph. Simply the photograph of the chromosome during the metaphase is called as the pictomicrograph. So after the pictomicrograph or the taking the photo, we arrange the chromosomes according to the length. According to the length of the chromosomes. Look at this structure. This is the one chromosome. This is the very long or the largest chromosome, and this is the 2, this is the 3, this is the 4th, 5th, 6th. That means from the higher length to the lower length we can arrange, and this arrangement of the chromosomes according to the length or from the higher, from longer to the shorter to the shorter to the longer, and the process, and it is called as the ideogram. Ideogram, or we can say that this is called as the karyogram. So what is the karyotype? What is the ideogram? Ideogram or the karyogram? You can already got. That means the ideogram is the simply the arrangement of the chromosome according to the length of the chromosome. Again, from the ascending and the descending, that doesn't matter, but this arrangement according to the length of the chromosome is called as the ideogram, and the karyogram after the pictomicrogram is called as the ideogram or the karyogram. So what is the karyotype? Simply this all the set of the chromosomes in any organism is called as the karyotype. Okay, so what is the significance of the karyotype? So after because of the karyotype we can eliminate or we can see the evolutionary relationship. So evolutionary relationship, for example, the chimpanzee. The chimpanzee has the nearly 98% similarity of the chromosome to that chromosome that is present inside human being. That means the chromosome, the human being and the chimpanzee have the similarity of the 98%. That means it can be concluded that the human being can be descended from the chimpanzee. So according to that, the karyotype, it helps in determining the evolutionary relationship between different organisms. So we have, for the human being, have the 46 chromosomes, or the 23 pairs of the chromosomes are present. 23 pairs chromosomes. So out of 23 pairs of the chromosome, 22 pairs are called as the autosome, and rest one pair is called as the allosome, or you can say that it is the sex chromosome. Okay, so this is the 22 chromosome: 1 2 3 4 5 6 7 8 9 10 11 12 13 14 15 16 17 18 19 20 21 22. So these 22 are called as the autosome, and this one is called as the allosome or the sex chromosome. That means this chromosome determines the sex or the human being. It determines whether the human being is going to the male or the female. It determines by the sex chromosome. Look at this structure. This is the female sex chromosome and this is the male sex chromosome. So that means if the human being is the male, so that means this will be absent. So if the female human being is present, that means this will be absent. That means only this one is present in the female. If it is the male, then this sex chromosome is present and this female one is left or the absent. So this is the main karyotype structure. So now that again another type of the chromosomes are present. This is called as the giant chromosomes. So what is the giant chromosomes? As the name suggests, the giant, that means a very large chromosomes are present. So two types of the chromosomes are present, which are present like the giant or the large structure is present. One is called as the polytene chromosome, and another is called as the Lamprey chromosome. So what these two chromosomes, that means the polytene and the Lamprey chromosome, they are present in the giant or the very large structure present. So let's suppose this is the chromosome, this is very tiny one or the teeny one, but this one, look at this structure. So this one is very large or the giant structure, and this one is called as the polytene structure. So the polytene chromosome is present in the dipteran. What is the dipteran? The dipteran, it is the order of an insect. That means the insect which has two wings. For example, the Drosophila, Chironomous. Please remember this one, Chironomous. It is in the NEET exam, it is always be asked that in the Chironomous. So again the fly. So these are the dipteran insects. So in these insects, in the salivary gland of the insect, these polytene chromosomes are found, or the polytene chromosomes are present. Okay, so this is the polytene chromosomes. So look at this structure. That means here the diameter is 20 micrometer, while in the length is the 25 millimeter. That means the length is very high. So this is the length, this is the width of the diameter. The diameter is the 20 micrometer, but the length is very huge, which is the 0.5 mm or the millimeter, which is very large of the giant structure. So this is the polytene chromosome which was discovered by Balbiani. So that's why these structures are called as the Balbiani ring structure. Balbiani ring, because the name suggests from this discoverer, which is the Balbiani in the 1881. In the 1881, Balbiani discovered this chromosome from the salivary gland of the dipteran insects. So in the Balbiani ring, in the polytene chromosome, look at the structure. This is the chromosome 3. This one is the chromosome 2. This is the chromosome 2 large arm. So this is the left arm. So this is the chromosome 4. That means many chromosomes are fused together, and now it forms the polytene chromosomes. So the polytene chromosomes, they are formed by the fusion of the many chromosomes, and now it can form the large of the giant chromosomes like structure it can form. So this is the polytene chromosome. Another is the another is the brush. What is the difference between this Lamprey chromosome? So the Lamprey means it is found mostly in most animals except mammals. That means mammals don't contain this Lamprey chromosome. Otherwise, maximum of the most of the animal, it contains the Lamprey chromosome. So in the Lamprey chromosome, or it was discovered by Fleming Fleming and Rocket in 1882, just after next year. In the 1881, the polytene chromosome were discovered by the Balbi, and in the next year, in the 1882, the Fleming and the Rocket discovered the Lamprus chromosome. In the most of the animals, but the mammals don't contain the Lamprus chromosome. So, what is the Lamprus chromosome? Simply, it forms by the two homologous chromosomes. Two homologous chromosomes. So look at this structure. These are the two chromosomes. They are the homologous to each other. So if we zoom this, it looks like this. That means this is called the loop-like structure. So this is the all about the polytene and the Lamprus structure, and these Lamprus and the polytene chromosomes, that the very giant or the large structure which is present in the polytene chromosomes. They are present in the diapteran animals, that means the insects, and the Lamprus, it contains in it is found in the most animals except the mammals. So this is all about the chromosome structure. If you like the video, please like and subscribe my video. Thank you.